Hi, this is JBPralay. Now we will know what is DNA. In briefly, DNA is a nucleic acid and essential for all forms of life. Nucleic acids are the organic materials present in all organisms in the form of DNA or RNA. The nucleic acids are formed by the combination of nitrogenous bases, sugar molecules and the phosphate groups that are linked by different bonds in a series of sequences. DNA stands for deoxyribonucleic acid, is a molecule that contains the instruction an organism needs to develop, live and reproduce. These instructions are found inside every cell and passed down from parents to their children. DNA is found in the nucleus, which is a small amount DNA also present in mitochondria and the eukaryotes. Mitochondrial DNA. The mitochondrial DNA is inherited from the mother to the child. The human approximately 16,000 base pair of mitochondrial DNA found. Similarly, plastids have their own DNA and play an essential role in photosynthesis. Discovery of DNA. DNA was first recognized and identified by the Swiss biologist Johannes Friedrich Miescher in 1869 in WBC. Mitochondrial DNA both found in plant and animal. The mitochondrial DNA is inherited by only mother, because it comes from mother cells of egg cell. Mitochondrial DNA we found in mitochondria. Mitochondrial we call powerhouse because it is produced energy at ATV and energy are produced. Mitochondrial DNA is a circular. DNA there are present 37 genes and it has R1 a non-coding region. Non-coding regions means not forms protein and does not occurs any function. In 32 genes to 22 genes are transformed to transformation. 2 genes are forming mRNA and 13 genes are forming proteins that are using NAD, ATP. NAD proteins are energy producing. A cell in human body may have 10 to 200 of mitochondria and each mitochondria with they are have hundreds of DNA molecules or more. In human liver cells have 1000 mitochondria, that means 1000 times 100 equals 1 lot to more DNA molecules. It may be varies, is it is average count. A mitochondria has 100 to more of DNA molecules and more. Nuclear DNA it is found in nucleus. Nuclear DNA inherited by both parents, mother and father. Nuclear DNA linear shape. In human body 3.2 billion base pairs of nuclear DNA and have 46 of chromosomes, which is 23 pairs in nucleus out of human body or human genome. Nuclear DNA 20,000 genes present in nucleus. To understand DNA, we need to know the some terms and basic structure and function. Let's start. Nucleic acids are biopolymer, macromolecules, essential to all known forms of life. They are composed of nucleotides, which are monomers made up of 3 components, which are pentose sugars, phosphate sugar, nitrogenous sugar. If the sugar is a simple ribose, the polymer is RNA, ribonucleic acid. If the sugar is derived from riboses as the deoxyriboses, the polymer is DNA, deoxyribonucleic acid. History of nucleic acid. Nucleic acid was first discovered by Friedrich Miescher in 1869 and he is gave first name nuclein. In 1889, Richard Altman creates the term nucleic acid as it has acidic properties. In 1953, Watson and Crick proposed the double helix structure of DNA. Occurrence and nomenclature. The term nucleic acid is the overall name for DNA and RNA member of family of biopolymer and is synonymous with polynucleotides. Nucleic acid were named for their initial discovery within the nucleus and for all presence of phosphate groups. Nucleic acid were first discovered within the nucleus of eukaryotic cell. These are two types of nucleic acids: ribonucleic acid, RNA deoxyribonucleic acid, DNA. Nucleosides. Nucleoside is the structural subunit of nucleic acids such as DNA and RNA. A nucleoside composed of nitrogenous base is either a pyrimidine, C, T, U, or purine, A or G, a 5-carbon sugar that is pentose sugars, which is either ribose or deoxyribose. Nucleotides equals nucleotide plus phosphate group. Nucleotides. The monomeric unit of nucleic acid are called nucleotides. Nucleic acids are also called as polynucleotides as it is formed by combining nucleotides. Nucleotides are phosphate esters of nucleosides, and nucleotides are made up of 3 components: nitrogenous base, pentose sugar, phosphatic acid. Nitrogenous base. Nitrogenous bases are heterophilic planar and relatively water-insoluble aromatic molecules. There are 2 general types of nitrogenous bases in both DNA and RNA, that is, 1 purine, 2 pyrimidines. The main function of nitrogenous base is to bond nucleic acid together. Purines. Purines is a heterocyclic aromatic organic compound that consists of a pyrimidine ring fused to an imidazole ring. Purine is water-soluble. Molecular formula C5H4N4. It is an anticlockwise. Adenine and guanine are called as purines. Adenine, 6-aminopurine, and guanine, 6-oxy-2-aminopurine. These 2 bases are found in both DNA and RNA. Adenine. Adenine is one of the 2 purine nucleobase used in forming nucleotides of the nucleic acid. In the case of adenine, the amino group attached at the 6th position of the purine, then it is called as 6-aminopurine. Guanine. Guanine is the one of the 2 purine used in forming nucleotide in the nucleic acid. Molecular formula C5H5N50. In the case of guanine, 1 oxy group is attached at the 6th position and 1 amino group is attached at the 2nd position, then it is called as 6-oxy-2-aminopurine. Pyrimidines. Pyrimidine is a heterocyclic aromatic organic compound similar to benzene. Molecular formula of pyrimidine is C4H4N2. Pyrimidine contains thymine, cytosins and uracil. Thymine. Thymine is one of the 3 pyrimidine nitrogenous bases used in forming nucleotides. In the case of thymine, 2 oxy group attached to 4th position of pyrimidine and 1 methyl group attached at 5th position, so that it named as 5-methyl-2,4-deoxypyrimidine. Molecular formula of cytosine. Cytosine is one of the nitrogenous bases that included in pyrimidine. In the case of cytosine, 1 oxy group is attached at 2 position and 1 amino group is attached at 4 position, so that it named as 2-oxy-4-aminopyrimidine. Uracil. Uracil is a nitrogenous base that replaces the thymine in the case of RNA. Molecular formula C4H4N2O2. In the case of uracil, only oxygen group is present in 2 and 4 positions, so it named as 2,4-dioxypyrimidine. Pentose sugar. Nucleic acids have 2 types of pentose sugar, that is ribose and deoxyribose sugars.
carbon atoms of the ribose and deoxyribose present in nucleoside or nucleotides are designated with a prime mark 1. Ribose sugar. Ribose sugar is present in case of RNA. It is a 5-carbon monosaccharides with a hydroxy groups on each carbon. Deoxyribose. Deoxyribose sugar is found in DNA. It is a 5-carbon monosaccharide lacking of 1 oxygen atom at 2' position. The hydroxyl group at 2nd position of ribose sugar is replaced by a hydrogen. Phosphate group. The acidic nature of nucleic acid is due to the presence of phosphoric acid. Nucleotides can have 1, 2, or 3 phosphate groups. Nucleotides with 2 or 3 phosphate groups are good energy donors. Phosphate groups are joined together to form phosphodiester bond. Phosphate groups can be also joined to other molecules as sugar. Multiple phosphate group have a strong tendency to repeat each because of the high concentration of the negative charge. When suitable enzymes are present, these repulsive force derives that transport to phosphate groups to other molecules with some energy being absorbed by as heat. Nucleosides, ATP and GTP are used not only for RNA and DNA synthesis, but also as energy donors for many cellular creations. Structure of nucleotides. Nucleosides. The sugar and nitrogenous base combination is called nucleoside. The bond between the sugar and the nitrogenous base is called the glycosidic bond. This bond is said to be in the beta of configuration with respect to the ribose sugar. The base is free to rotate around the glycosidic bond. Due to the rotation of the glycosidic bond, 2 different conformations are possible. The 2 standard conformations of the bases around the glycosidic bond are syn and anti. Pyrimidine tend to adopt the anti-conformation A most exclusively because of steric interference between O2 and cis in the syn conformations, whereas purine are able to assume both forms. Nitrogenous base. Adenine. Guanine. Cytosine. Thymine. Uracil. Nucleosides form adenosine, guanosine, cytidine, thymidine. Nucleotide forms adenylic acid, guanylic acid, cytidyllic acid, thymidylic acid, uridylic acid. Polynucleotides are polymers of nucleotides, which are formed by condensation of nucleotides. The condensation reaction occurs between the alcohol of a 5' phosphate at 1 nucleotide and the 3' hydroxyl at 2nd with the elimination of H2O. The linkage between 2 nucleotides is known as phosphodiester bond. The phosphatases of polynucleotides are acidic, so, at physiological pH, polynucleotides are polyanions. All nucleotides in a polynucleotides chain have same relative orientation. The formation of phosphodiester bonds in DNA RNA exhibits directionally. The primary structure of DNA or RNA proceeds in the 5' to 3' direction. First we will go to the structure of DNA. The double helix structure of DNA was discovered by James Watson and Crick in 1953. Double helical structure of DNA. The structure of DNA is a double helix structure because it looks like a twisted ladder. The sides of ladder are made up of alternating sugar deoxyribose and phosphate molecules. The steps of the ladder are made up of a pair of nitrogenous bases. There are 4 types of nitrogenous bases. Have a specific pairing. This pairing pattern, of course, because the amount of A equals to the amount of thymine, the amount of guanine is equals to the amount of cytosine. The pairs are held together by hydrogen bonds. DNA is a double-stranded helix, that is, each DNA molecule is comprised of 2 biopolymer strands. coiling around each other to form a double helix structure. These two DNA strands are called polynucleotides. As each strands they are made up of simpler monomer units called nucleotides. Each strands are anti-parallel, meaning that one strand runs in 5' to 3' direction, while the other strands run together by 5' directions and are complete to each other. Basically, DNA is composed of deoxyribonucleotides. The deoxyribonucleotides are linked together 3' to 5' means phosphodiester bonds. The nitrogenous bases that compose the deoxyribonucleotides include adenine, guanine, cytosine, and thymine. AT complementary of the strand via this 2 hydrogen bonds on the opposite strand. The shape of the helix is stabilized by the hydrogen bonding between bases. In a double-stranded DNA, the number of A bases are equal to the number of T' bases and G bases are equal to C'. One of the strands in a double helix is known as sense strand, that is, which codes for RNA, proteins. Another strand is known as antisense strand. The length of each interval is 34 nanometer and diameter is 20 Armstrong/2 nanometers. The distance between each turn is 3.4 nanometers. There are 10 nucleotides per turn. As a result of the nature of DNA, the molecule has 2 asymmetric grooves. One is major or other one is minor grooves. Grooves for binding of regulatory proteins. The minor groove of ATRAC DNA provides a unique chemical environment. The polarity and electronegative potential are high. The floor and walls of the groove are lined with hydrogen bond acceptors but are devoid of hydrogen bond donors. There are 3 different types of DNA. A DNA it is right-handed double helix similar to the B DNA form. Dehydrated DNA takes in a form that protects the DNA during extreme condition, such as dissociation. Protein binding also removes the solvent from DNA and that takes in a form. B-DNA. This is the most common DNA conformation and it is a right-handed helix. Majority of DNA has AB-type conformation under normal physiological conditions. Z-DNA. Last one is Z-DNA. It is left-handed DNA where the double helix winds to the left in a zigzag pattern. It is found ahead of the site of a gene and hence is believed to play some role in gene regulation. Properties of DNA. DNA helixes/helices can be right-handed or left-handed. DNA having the right-handed is most stable. On heating, the 2 strands of DNA separates from each other and on cutting there again hybridize F. The temperature of which the 2 strands separates completely is known as melting temperature. Melting temperature is specific for each specific sequence. The sample of DNA having higher melting point must have more C triple bond G content because C triple bond G pair has 3 hydrogen bonds. The sequence of bases along the DNA molecule encodes for the sequences of amino acids in every protein in all organisms.
Thank you for watching. Please like, share and subscribe my channel.